The topic that we shall be discussing is about virus-host interaction. It is the interaction between the virus and the host. That is, as we have already discussed, there should be a specificity between the host and the virus. Only then there can be attachment of the virus to the host cell and initiation of the infection. So in this particular topic, we'll be dealing about how the virus interacts with the host and causes the pathogenic effect in the host. So coming to the introduction part, the virus-host interaction can be studied at the three levels. First is the cellular level, next is the individual level, and followed by the community level. So in this particular topic, we'll be dealing about the cellular level of host viral interaction. So these organism viruses, which are obligate intracellular parasites, have the ability to cause much damage at the cellular level. They produces broad spectrum of diseases which can range from no apparent infection to the most profuse damage to the host cell. This can be very well studied in case of the poliovirus, which is usually cytolytic or cytocidal in nature. That is, after the synthesis of the progeny virus, it has the ability to lyse the host cell. But in the case of other viruses, you can see that there is no damage that is caused to the animal viruses or the human cells. Whereas in the case of bacteriophages, there is a lysis of the host cell, that is the lysis of the bacteria and release of the progeny. But in case of animal viruses, the profuse damage is mostly seen in the case of polio viral infections. And here the polio virus is said to be cytocidal or cytolytic in nature. Next is there are certain viruses which have the ability to cause the cellular proliferations. This can be very well studied in molluscum contagiosum, and some oncogenic viruses have the ability to produce malignant transformation in the host cell. Apart from that, there is a possibility that certain viruses will have the ability to live in harmony with the host cell. Here, in the case of this particular phenomena, which is called as a steady state function, both the host as well as the virus replicate independently without causing any effect to each other. So this particular phenomena does not cause any type of damage either to the host cell or to the virus, which is usually multiplying inside the host cell. And the effect of the viruses can be very well studied in the tissue cultures, where the virus usually brings about the morphological changes in the tissue culture. So this can be studied with the help of cytopathogenic effect. So this cytopathogenic effect may not be parallelly similar in the animal inoculation, because there are certain defense mechanisms that comes forward to eliminate the viral infection, and there is completely a different clinical picture of the cytopathic effect that are brought about in the animal or the human host cells. So the cellular damage that is caused is very severe in case of certain type of viral infection, and there are many factors that usually determine the cellular damage that is caused during the viral infection. The first and foremost being the proteins that are synthesized after the viral nucleic acid usually gains an entry into the host cell. So first, when the virus gains an entry into the host cell, it releases the nucleic acid, and this nucleic acid takes over the host cell. So during this process, there is a transcription of mRNA, which usually synthesizes the early or the non-structural proteins. Now these non-structural proteins, when they are synthesized, there are certain regulatory proteins that are synthesized by the viruses, which completely takes over the host cell. Now this is a sort of damage to the host cell, because the host cell will not be able to multiply in a normal way as we can see without the viral infection. Apart from that, when there are progeny virus that are synthesized inside the host cell, there is a morphological changes that are seen in the host cell, ultimately ending up to the toxic effect to the host cell. And next type of cellular damage is the damage to the plasma membrane. When there is a damage to the plasma membrane, there are certain enzymes that are released by the host cell that usually causes the autolysis or the self-destruction of the host cell. This is another sort of cellular damage. Next, coming to the cytoplasmic membrane, there are certain viruses that have the ability to fuse the cytoplasmic membrane of the two cells. That is called as a polykaryocytosis or a syncytial formation. Example is the respiratory syncytial virus, which have the ability to form a multinucleate giant cell in the media which are usually cell culture technique. Here you can see or identify the growth of the organism with the help of the multinucleate cell or the syncytial formation. And apart from that, the viruses, after multiplying and releasing its progeny, viruses expresses certain types of antigen on its surfaces. So these antigens are usually called as hemagglutinins, and these hemagglutinins have the ability to agglutinate the RBCs, and hence they are called as heme adsorption. So identifying the heme adsorption is best method to identify the viral infection that is caused to the cell. But there is damage to the RBC, where the RBCs usually agglutinate in the presence of the viral hemagglutinins. And certain oncogenic viruses, as we know, when they gain an entry, they completely transform the cells, and it is a host cell that will have the ability to express the viral antigen. So there is a complete morphological change that can be seen during an oncogenic infection. So these are the different cellular damages that are usually seen when there is an infection to the host cell by the viruses. Apart from these infections, certain viruses also have the ability to interact with the genetic material and cause damage to the genetic material. So this is very well studied in case of adenoviral infections, and which has been demonstrated in vitro. They are thought to produce chromatid grafts and a breakage in the chromosome 17 by the adenovirus 12 and 35. So apart from damaging the cellular material, the organism also have the ability to interact with the genetic material and cause a damage to the genetic material. And these are about the cellular damage. But keeping aside this cellular damage, we can see that there are certain viruses which have the ability to produce certain types of inclusion bodies during their replicative cycle, or during their synthesis of the progeny virus, or during their metabolic activities. So these inclusion bodies are of various sizes, shapes, they have different morphology, they have different staining properties, and the presence of these inclusion bodies in different areas of the body helps in identifying the strain of the viruses. So for example, the pox virus have the ability to produce these inclusion bodies in the cytoplasm, whereas the herpes can produce these inclusion bodies in the nucleus. So depending upon the location where these inclusion bodies are present, you can identify the characteristic of the virus or the strain of the virus that is involved in the pathogenesis of the disease. And in case of measles, you can see the inclusion bodies present both in the cytoplasm as well as in the nucleus. And these inclusion bodies are usually acidophilic in nature, and you can stain them very well with the help of Giemsa stain or eosin methylene blue, and they usually stain pink in color, which can be very well identified under the light microscope. So most of the organism produces the acidophilic granules, inclusion bodies you can say, and it is adenovirus that have the ability to produce the basophilic inclusion body. So based upon the staining property, you can identify whether it is an acidophilic granule or a basophilic granule, and depending on that you can identify the viral species. Next is there are certain viruses that have the ability to produce intracytoplasmic eosinophilic granules. These are called as the Negri bodies that can be seen during the rabies infection, and these Negri bodies are usually present in the brain cells of the animals or the host cell. So identification of the Negri bodies helps us to identify the rabies infections. And apart from these inclusion bodies, you can also see multiple inclusion bodies which are called as guarneri bodies produced by the vaccinia virus. And Bollinger bodies are produced by the Fowlpox virus, and the molluscum bodies are produced by the molluscum contagiosum. And based upon the type of inclusion bodies, Cowdrey in 1934 has classified these inclusion bodies into two major types. One is a type A, and the other one is a type B. Type A is the one which has variable sizes and it is granular in nature, and mostly seen in the case of herpes and the yellow virus, whereas type B usually are circumscribed, or they have a particular accurate size you can say, and apart from that there are multiple granules that are produced, inclusion bodies that are produced during this infection. And the examples of the type B are adeno and the polio virus. Irrespective of whatever the inclusion bodies that are produced by the organism, they bring about degenerative changes in the cell which completely changes its staining properties. So with the help of staining technique, we can identify whether it is is a normal cell or a virus infected cell. This is about the interaction, how the virus usually multiplies inside the host cell and produces different types of material with which you can identify the viral infection. Now coming to the pathogenesis of the viral infection, how it usually gains an entry and how it usually causes the infection. So based upon the clinical outcome of the infection, we can classify the viral infection into two major types. One is the inapparent or subclinical, where you will not be able to see the symptoms immediately, or it takes very long time to see the infection. Next is apparent or clinical or overt. There are three terminologies that are given to this infection: apparent, clinical or overt, which are further subdivided into acute, or it is a subacute or chronic infections. So apart from these two infections, there are certain viruses that can become latent inside the host cell and causes latent infection. So we shall be dealing in detail about the latency of the viral infection and how and which virus usually causes the latent infection. So depending upon the incubation period, you can classify the latent infection into various types. First is the recurrent herpes simplex or herpes zoster infection. Here in the case of this particular infection, there is a long period of quiescence, after which you will be able to see the symptoms. And in this particular case of infection, the organism have the ability to multiply in the nerve root of the ganglia. That is, it keeps on carrying out its normal metabolic activity without causing any symptoms, but it takes a prolonged period of incubation period to express its symptoms. So it is called as the recurrent herpes simplex or the herpes zoster. So here we should remember that there is a primary infection, after which the organism becomes latent and shows the latent infection later on after certain months or periods, it can take up to years or to show up the infection. Next is a persistent tolerant infection. So here in the case of persistent tolerant, I mean to say that there is an immunotolerance that has been developed towards the viruses, as a result of which there is no immune response that is developed and there is no pathogenic effect of the virus on the host, mostly seen in the neonatal infections. And the best example that can be seen in the case of persistent tolerant infection is the lymphocytic choriomeningitis seen in the mice. So here there is a tolerance that is developed towards the virus. Once the tolerance breaks, then you can see the development of the infection and the outcome of the infection, that is the clinical symptoms that can be clearly apparent. Next type of latency that can be seen is a slow progressive. The name itself indicates that the incubation period is very long, and the example in this is in the case of the disease called as scrapie, which is seen in the sheep, and kuru in the case of humans. So here the latency of the virus is for a very longer period of time, and it can take years together to show up the clinical symptoms. Next latency is by the oncogenic viruses. Here it takes about nearly months or years together to show up the symptoms, and this depends again on the immunocompetency of one individual. That is, whether the person is he is very healthy or very weak in his immune system. So depending upon that, the viral oncogenic virus usually shows the latency and appearance of the symptoms. And the best and the last and the best you can say is the latency given by the HIV virus, where initially you can see progressive or symptomatic diseases followed by the progressive damage to the immune system, which later on becomes very fatal in nature, and this is usually expressed after many years. So this is about the latent infections that the organism usually causes. So apart from clinical and subclinical infections, there are certain viruses that can become latent inside the host cell and can show up the symptom after many years together. Now next, coming to the entry of the viruses into the host cell, the entry of the viruses can be you can say there are different portal of entries by which the virus can gain an entry into the human and cause the infection. So the most important being the respiratory or the alimentary tract, by which they can gain an entry. Next is the skin. The virus can also gain entry via the conjunctiva or the genitals, and the last can be the vertical transmission from the parent to the progeny. So apart from the other barriers, there is a transmission of virus from the parent to the progeny mostly during the delivery process. Coming to detail in about the different types of portal of entry, let us first discuss about the respiratory tract by which the virus can gain an entry. As we are in constant association with the different types of microorganism, there is most probability that the virus can gain an an entry via the respiratory tract. So it is the most suitable and the most favorite spot for the entry of the viral infection. So here when the virus gains an entry, it multiplies locally for certain period of time, which is usually called as a silent local infection, after which the virus can get disseminated or can be carried out via the lymphatics or via the bloodstream towards the various part of the organs of the body, and where they can show the active multiplication. So when they reach the target site, the organism will have the ability to multiply effectively and show the pathogenic effect by producing the lesion. So it is the portal of entry, that is a respiratory tract, for which there is an initial multiplication, after which there is a dissemination of the virus to the various parts of the body. Once they reach the target organ, they will have the ability to actively multiply and show up the pathogenic effect. This is in the case of systemic infection, but in case of certain respiratory viruses such as influenza and the rhinovirus, the multiplication is only local, only at the respiratory tract. Only the cells of the respiratory tract are infected and they show the pathogenic effect. And hence here in the case of this particular infection, which is a respiratory infection, you can see a short period of incubation, whereas in the case of systemic infection, the incubation period can be for a longer period of time than the local infections. Next important portal of entry is the alimentary tract. But here one thing that should be remembered is most of the viral will be eliminated in the intestinal tract due to the digestive juices that are usually secreted. Only the organism which will have the ability to attach to the cells of the intestine will have the ability to multiply and cause the infection. Otherwise, most of the viruses will be eliminated. In case of rhinoviruses, they are highly susceptible to the gastric acidity, and hence these rhinoviruses will not be able to cause infection in the enteric part of the human body. And it is only gastroenteritis virus such as hepatitis B that will have the ability to cause the infection. And the most actively growing organism in the gut is a rotavirus. And coming to the polio virus, this is an enterovirus, but shows its effects in the central nervous system. So it multiplies locally in the intestine and will have the ability to get disseminated through the various like you can say lymphatic or the bloodstream and cause the like infection of the central nervous system, where in the case of this poliovirus you can see the negri bodies in the brain of the human. So apart from that, you can identify the growth of the organism in the various parts, but the major or the satisfactory position where these organism multiplies is in the enteric part of the human body. Next is the skin, the most important portal of entry. This being the third, you can say the gain of entry for the viruses, as viruses are obligate intracellular parasites and they cannot usually survive in the external period for a longer period of time. So immediately they have to attack the cell. So by the time they are in the external environment, most of the viruses will get eliminated. But one viruses which have the ability to gain an entry through the skin is the papilloma virus, and they usually produces the local lesions. Apart from papilloma virus, we have vaccinia, cowpox virus, which usually shows the dermal lesions, and these are the characteristic feature of this particular organism. And this skin infection are said to be the exanthemus viral diseases which are secondary to the systemic infection. And the virus can gain an entry and cause the infection depending upon the site of entry. Like for example, when there is an abrasion, the most suitable area for the growth of the virus is the papilloma virus. So this papilloma virus gains an entry via the abrasion. Next is the arbovirus. Arbovirus usually are carried by the insects, and by the insect bite, these arbovirus can cause the infection, which are mostly the systematic infection. Next is hepatitis virus, which is usually transferred via the contaminated injections. And in case of the systemic spread, the very well studied example is the rabies, with the help of the dog bite. So when the dog bites, the organism gains an entry locally and then reaches the spinal cord and can go to the central nervous system. So this is the systemic spread that is caused by the rabies infection. Next, another important portal of entry is the conjunctiva, which is very rarely seen, but there are certain organisms that will have the ability to cause the infection of the conjunctiva. So the local diseases are caused by arbovirus, and the systemic spread is mostly by the measles. And apart from these portal of entries, there is another type of entry, that is a genital tract, where the sexual contact can lead to the transmission of infection. The example is the HIV. And next is the vertical transmission between parent to progeny. So there are congenital infections that can be caused ranging from the development of the ova to the birth of the child. So there can be infection that can be transmitted by the infected mother. So they can be acute systemic infections that are caused like rubella and cytomegalovirus, and the vertical transmission of the tumor viruses can also take place, and in the case of avian leukosis virus, the transmission is via the ova, and in the case of murine memory tumor virus, the transmission is through the breast milk. So it is the transmission of the virus from the parent to the progeny. Now next, coming to another important topic, that is spread of the virus in the human body. Once the organism have the ability to attach and like has the ability to gain an entry, next is how these viruses usually spread in the body and causes systemic infection. So as already discussed, there can be two type of infection. One is a local infection, the other one is a systemic infection. In the case of local infection, at the immediate site of gain of entry, they have the ability to multiply and show of their pathogenic effect. But in case of systemic infection, they have to gain an entry through the lymphatics or the blood streams to reach the target organ and show the pathogenic effect. So here we'll see the spread of the virus. So this was very well demonstrated by Fenner in 1948 using mousepox virus. So here he was able to demonstrate how the virus usually gets disseminated inside the human body and causes systemic infection. So in the case of mousepox virus, the virus usually gains an entry via the skin, and through the skin the organism is taken to the lymphatics, where they reach the lymph nodes, and here in the lymph node they actively multiply and disseminate the virus into the blood. So this is called as a primary viremia. Now when the primary viremia takes place, the viruses are carried on to the spleen or the liver, where they usually multiply again for the second time, giving a central foci. Now after the central foci is formed, here again the virus gets disseminated into the blood. This is called as a secondary viremia. Now it is this secondary viremia that carries on the viruses to the target organ, where the you can see the pathogenic effect of the virus in the form of lesion. So this is how the viruses usually gets disseminated into the body and causes systemic infection. So this is the model that describes how the viruses usually disseminate. So similar type of study is been seen in the humans, where the dissemination of the virus takes place to cause the systemic infections. Coming to the incubation period, the duration by which there is a gain of entry of the virus to the production of the symptoms is called as the incubation period. So the duration of the incubation period depends upon the three type of relationship. One is the site of entry, next is the multiplication and the formation of the lesion. So as already discussed, when this virus gains an entry and replicates locally and produces a pathogenic effect, there is a short incubation period. In case of influenza virus, as the organism gains an entry via the respiratory tract and is limited to that area to get multiplied and show the pathogenic effect. So in the case of this type of infection, there is a short incubation period. But in case of systemic infection, where the organism has to get disseminated, carried on via the lymphatic and the bloodstream to the target organ to show the pathogenic effect, in that case you can see a longer incubation period. There is another you can say a slight variation over here. In case of arbovirus, which have the ability to introduce the virus directly into the bloodstream, there is a very short incubation period of 5 to 6 days. And in case of respiratory and the gastroenteritis, the incubation period is very short. And in case of hepatitis, it takes about 2 to 6 months. And the papilloma and molluscum contagiosum are the viruses which has a very longer incubation period of time. Next, coming to the host response to viral infection. Now here in this particular topic, we'll be seeing how it is not only the virus that brings about the clinical outcome of the disease, but it is a resistance of the host. So it has to be like eliminated from the body easily in order to exclude the virus from causing the infection. So it is not only the virus that causes the infection, but the other factor is also very important to show the clinical outcome of the disease, and it is a resistance of the host. If the person is highly like shows a high immune status, then the pathogenic effect of the virus will be inactivated. Whereas the person with who is immunodeficient, you can see that they are more prone to different types of viral infection. So the virulence of the organism is completely dependent not only upon the virus but also the resistance of the host. And coming to the resistance of the host, we will broadly classify into two types. One is the immunological and the other one is the non-specific immune response. So as we have seen in the immunity, there are two types of immunity. One is the innate immunity and the other one is the adaptive immunity. So innate immunity is a non-specific immune mechanism. It is not specific to any type of bacteria or viral infection. Whereas in the adaptive immunity, there is a specificity towards either it is a bacteria or the viral infection. So the resistance of the host can either be immunological, that is an adaptive immunity. Next, it can be the non-specific immunity. Irrespective of whatever it is, both of them combine together to give a positive effect in the elimination of the infection. So here in the non-specific immunity, we will be studying about the genetic and the physiological factors, the interferons, which are the antiviral substances that are produced by the human body in response to the virus, the body temperature, the nutrition and the hormones. First, we'll deal about the immunological aspect of the host-viral interaction. So viral infections and how the immunity usually combats this viral infection. So virions are nothing but actively infecting virus particles, or the infectious virus particle is called as a virions, and they act as a very good antigen. Now, as we know that immunity usually deals with the production of or enhancement of the immune status towards the foreign bodies, that is the antigen. So in response to the viral antigen, human host produces different types of immune response, that is the humoral immune response and the cellular immune response. In case of humoral immune response, it is antibody mediated, and the antibodies that are generated are highly specific towards the viruses. And during the course of the immune response, you can see the production of different types of immunoglobulins, usually IgG, IgM and IgA. Apart from which, you have two other antibodies or immunoglobulins that are not involved in viral infections, which are IgE and IgD. But in the case of viral infection, it is IgG, IgM and IgA. And in response to the viral infection that is coming via the blood or the tissue spaces, IgG and IgM are highly effective to eliminate the organism. But in case of the mucus secretions, where the virus have the ability to multiply, the IgA is the antibody that comes forward to eliminate the infection. So irrespective of whatever the immunoglobulin is, the effective part of the immunoglobulin is to neutralize the virus. So here in the case of bacterial infection, we can see that there is completely killing of the bacterial particle, but here in case of viral infection, it is neutralization of the virus. I mean to say, neutralization means they will make the virus inactive to bind to the host cell receptor, as a result of which when there is no attachment, there is no release of the nucleic acid and there is no infection of the virus. So the antibodies that are generated against the viruses are called as neutralizing antibodies, similar to the antibodies that are produced against the toxins of the bacteria. So here we will not call them as antibodies, but we will call them as neutralizing antibodies. Apart from this, the other immune response that comes forward is the complement mediated lysis of the host, that is complement mediated lysis of the viruses. So here the complement proteins along with the antibodies will be able to eliminate the enveloped virions. And the antibodies that are generated towards the viruses can be of different types, because the antigen can either be an internal antigen or the surface antigen that are usually expressed on the viruses. So the antibodies that are produced against the internal antigens are not neutralizing, but whereas the antibodies that are produced against the surface antigens are usually neutralizing in nature. The surface antigens will study study with the help of influenza virus. The influenza virus expresses two types of spikes or the peplomers which are antigenic in nature. One is the hemagglutinin, the other one is a neuraminidase. So towards this hemagglutinin and neuraminidase, different types of antibodies are produced called as anti-hemagglutinin and anti-neuraminidase. But the effect of these two are completely different. In the case of anti-hemagglutinin antibodies, these antibodies will inhibit the attachment of the viruses to the host cell. But in case of anti-neuraminidase antibodies, they will prevent the release of the progeny virus. So hemagglutinin helps in binding of the virus to the host cell and initiate the infection. And now after the infection is done, the progeny viruses are synthesized. To release the progeny virus, they require the neuraminidase enzyme. So the antibodies are directed towards the neuraminidase and inhibit the release of the progeny virus. So there are two different types of antibodies that usually neutralizes the viral infectivity. Apart from that, these can enhance. There are certain sides. You can see, as we can see, every coin has two sides. So the immune response can also give a positive effect and a negative effect to the human host. So apart from protecting against the different viruses and bacteria, sometimes these antibodies also shows effective viral infectivity, that is, they enhance or exaggerate the viral infection. So they have the ability. Now this usually takes place in case of autoimmune diseases where there is a severe damage to the host cell. When there is a severe damage to the host cell, the immune response comes down, and as a result of which there are certain viral infection that gets exaggerated and cause severe or a profuse damage to the host cell. Example is we can see over here is a respiratory syncytial viral infection which usually takes place in the infant below 6 months. Now here the thing that was demonstrated was the antibodies which are passively transferred from the mother to the fetus usually protects the fetus or the child for the 6 months, after which they usually gets eliminated. Now during this 6 months, if the child is affected with the respiratory syncytial virus infection, there is more exaggerated damage to the child who are less than 6 months. Now after 6 months, if there is an infection of this particular virus, then it is very mild in nature. Apart from that, the antibodies can also cause the pathogenesis of the antibodies can cause viral hemorrhagic fever, which can be explained with the help of immune thrombocytopenia, and the extrahepatic lesion that are usually seen during hepatitis B infection are due to the damage that is caused to the cell by the immune complexes. So not only they protect the body from the different infection, but sometimes when there is low immune competency in an individual, you can see the antibodies aggravating or exaggerating the viral infections. This is about the humoral response. Now coming to the cell-mediated immunity, the importance of the cell-mediated immunity was very well understood with the help of delayed type of hypersensitivity reaction after the vaccination process. And it is said that in the case of a gamma globulonemics, that is a person who does not produce immunoglobulins, in that case the cell-mediated immunity usually comes forward to protect the person from the different types of viral infection. But here one thing should be known that this cell-mediated immunity does not provide infection when there is a viremia, but usually protects from the viral infection that causes localized infections as well as the oncogenic viral infections. And the decrease in the cell-mediated immunity or the loss of cell-mediated immunity can be we can know the importance of the cell-mediated immunity when the cell-mediated immunity has come down. You can see there is a recurrent infection of the herpes, pox and measles infections. Next is this cell-mediated immunity not only protects but also causes a damaging effect to the host cell, which can be seen in the tissue damage caused in the lymphocytic choriomeningitis. And apart from that, antibodies can also suppress the immune response, which can be very well studied in the case of measles. When there is a measles infection, this measles infection usually inhibits the delayed type of hypersensitivity reaction towards the tuberculin, and hence the underlying tuberculosis will not be able to be identified. Next is in case of lymphocytic choriomeningitis, this infection usually completely decreases the production of antibodies, and the very well studied example is with the HIV, which attacks the CD4 receptors on the T cell, completely bringing down the immune response, leading to secondary infections and ultimately leading to the fatal disease. Irrespective of whatever it is, when the virus attacks the human host usually produces a solid immunity and a lifelong immunity. And the exception is with the common cold or the influenza virus, because these organisms keeps on changing their antigenic character every year, as a result of which there are no vaccines that are produced against the influenza, and there is no immune response which can be a lifelong immune response towards this particular organism. Now, by the use of live viral vaccine, we can completely bring down the infections that are caused by these viruses, and the mortality rate can be brought down by use of live viral vaccines. Now, next, coming to non-immunological response, which is an innate immunity that comes forward to protect our body against the viral infections. Coming first to the phagocytosis, it is a process of engulfment of the foreign particle by the different cells of the immune system. The most important cells that are involved in phagocytosis are the macrophages and polymorphonuclear leukocytes. In the case of this polymorphonuclear leukocyte, they do not give a protective role against the viral infections, but in contrast to it, you can see that there is a decrease in the PMNLs, which is called as a leukopenia during the viral infection, and the only cells effective against the viruses are the macrophages, which will have the ability to attack or catch up the viruses which are present in the bloodstream and cause the phagocytosis of these viruses. Next, coming to the body temperature. Now, the body temperature, as we know that the viruses are highly heat labile in nature, getting inactivated within seconds in 56 degrees centigrade and in minutes at 36 degrees centigrade. So, the human body temperature will not allow the growth of the virus particle, and which can lead to pathogenesis. But there is an exception over here. In the case of herpes simplex virus, this organism have the ability to produce the fever blisters during the high temperature in the human host. And next is accompaniment of the herpes febris with the pneumococci or the streptococcal infections. Next important non-specific defense mechanism are the hormones. Hormones can also have the ability to either induce a viral infection or inhibit the viral infection. So the administration of the corticosteroid can enhance the viral infections, and the susceptibility of this Coxsackie virus B1 can be studied when the corticosteroids are usually given. And in case of varicella and vaccinia, which are mild infection, but if the person is on corticosteroid, they can show a profuse damage to the host cell. Next, the injudicious use of steroids can also enhance the viral infection. This can be very well seen in the case of herpetic keratoconjunctivitis, which can ultimately lead to the blindness. And the viral infection during the pregnancy can cause the most fatal diseases. During the pregnancy, there are variation in the hormone production. Now, it is the reason because of which the pregnant ladies are most prone to viral infection. The cortisone effect on the immune system is said to be that it usually inhibits the immune response, and the inability to produce the interferon towards the viral infection is because of the presence of the cortisone that ultimately leads to the enhancement of the viral infection. Next, coming to the malnutrition. Here, in the case of malnutrition, the most important studied disease is the measles. Here, in the case of measles, this measles has the ability to show its enhanced effect in the children who are malnourished. So, in case of well-fed babies, the measles can easily come down, and there is no much treatment that is required in the case of well-fed babies than to the malnourished children. Coming to the age, the two extreme ages are most prone to viral infection. The two extreme ages being the infants and the most old people, where you can see the immune response is very low in these particular ages. Now, coming to the next important topic, that is the interferons. These interferons are nothing but the antiviral substances that are produced in response to the viral infection by the host. So, they are natural defenses that are produced against the viral infection. This was first demonstrated by Isaac and Lindelman in 1957, where they were studying about the chick chorioallantoic membrane. When they challenged the chick chorioallantoic membrane with the live or the inactivated influenza influenza virus, they could see that this membrane produced an antiviral substance against the influenza virus, and hence they called it as antiviral substances. So now we know that these antiviral substances are produced as a natural defense against the viral infections. And interferons are mostly host-coded proteins, that it is the host that usually synthesizes these proteins in response to the viral infection. And the best thing to know about the interferons is it protects, it does not protect the cell that has been already attacked by the viruses, but instead it protects the cells which are adjacent to it. So already attacked cells produces the interferons, and these interferons in turn goes and protects the other cells and makes them refractory or resistant towards the viral infection. Now the function of this interferon is to produce translating inhibiting protein or translation inhibiting protein in the unaffected cells. So in response to the interferons, the healthy cell usually produces this particular protein, which is a translation inhibition protein. It the name itself indicates that it inhibits the translation of the viral mRNA. So as we know that in order to synthesize the proteins, the mRNA has to get translated. But here when these TIPs are usually produced, it inhibits the translation of the mRNA of the virus, as a result of which no proteins are synthesized and the virus usually gets inactivated. And this TIP is usually made up of three important components. One is the protein kinase, oligosynthetase, and the third one is the RNase. RNase have the ability to degrade the viral RNA. These interferons is said to be species specific. That is, the interferon produced by one species can protect only the closely related species but not the other species. And they are not virus specific. They are produced in response to any type of viruses and can confer immunity to any type of viral infections. And there are different viruses which shows variation in the susceptibility to interferons. And the viruses, there are different viruses that can have the ability to induce the production of the viruses. Some viruses are good inducers of the interferon production. Some viruses are poor inducers, you can say, in the case of interferon production. So the viruses which are cytocidal and virulent viruses are the poor inducers of the interferons, whereas the avirulent viruses are said to be a good inducer of the interferons. Now coming to the RNA and DNA viruses, RNA viruses are said to be a very good inducer of the interferons, whereas the DNA viruses are poor inducers of the interferons. And the potent inducers are usually the Toga virus, Sendai virus, or NDV virus. So whenever there is an infection with this virus, you can see more amount of interferons produced by the host cell. And the production of the interferon is dependent on the temperature. If the temperature is increased up to 40 degrees centigrade, there is a more potent interferon production that can be seen at 40 degrees centigrade. And the decrease of the interferon is dependent upon the steroids and the low oxygen tension. That is, at this particular factors, you can see that the interferon production usually comes down. And the synthesis of the interferon takes place within 1 hour of the viral infection. The peak of the production of interferons usually takes place between 6 to 12 hours. Now, coming to the antibody response and the interferon production. The production of the interferon is very quick when compared to the antibody response, and hence they are said to be more potent against the viral infection than the antibody response. So they are the first that comes forward to protect against the viral infection before the antibody are produced, or the neutralizing antibodies that are produced against the viruses. And the production of interferon depends upon the cellular transcription and the protein synthesis. Both of these factors are necessary in the host cell to synthesize the interferons. And there are different types of interferons that have been identified. There are three most important classes of interferons: interferon alpha, interferon beta, interferon gamma. So based upon the antigenic character, based upon the cell origin and the other factors, you can classify this into three major types. And they are usually written as IFN. That is the short form of interferon. And the prefix which is usually usually attached depends upon the species from which the interferons are usually produced. Like in case of HUIFN, it is a human from which the interferons are usually produced. So they are designated as IFN and they are nothing but the interferons. Coming to the alpha interferons, these are called as the leukocyte interferon. They are non-glycosylated and produced in response to the suitable viral infections, and there are 16 subtypes of this alpha interferon that are known to us. Coming to the beta interferon, they are called as the fibroblast interferons, and they are produced against the viral antigens or sometimes towards the polynucleotides, and they are glycoproteinaceous in nature. Coming to the gamma interferons, they are called as the immune interferons produced by the T lymphocytes, produced against the antigens or mitogen. They are mostly immunomodulatory. They are immunomodulatory and anti-proliferative activity, but they do not show any type of effect on the viruses, that they are not antiviral in nature. And these interferons can easily get inactivated by the proteolytic enzyme, but not by any other enzyme such as nucleases or any other enzymes. Because they are proteins in nature, they can be easily inactivated by the proteolytic enzymes, and they resist towards different types of infections that are caused by the viruses. They are non-specific in nature, and the molecular weight of this interferon is 17,000 daltons, and they are poor antigens, that is, they cannot induce an immune response in the other species when these are introduced into the other species. And because they are poor antigen, it is difficult for us to identify them with the help of serological test. And the best assay method to identify the interferon is called as the interferon assay, which is usually carried out in the cell culture technique by inhibiting the growth of the virus. Now, when there is a presence of interferons, the virus usually does not multiply, and hence there is no cytopathic effect that can be seen in the cell culture. So, based upon this, you can identify whether interferons are produced by the cells or not. The potency of this interferons is usually measured in the term of international unit per ml, and they are the ideal candidates towards the different types of viral infection. They are non-toxic and non-antigenic, but they have the ability to diffuse in the whole body, providing a systemic immunity towards the different types of viruses. Now, the drawback over here is it is species specific. As I told you, the interferons which are produced in one species is active only in the related species but not in the other species, and hence we cannot make use of these interferons that are produced elsewhere in the chick or the rabbits to protect against the human diseases. So the human interferons have the ability to protect against human viral infection and protects only to the humans but not to the other species. Variations have been brought about in the clinical laboratory to produce interferons from the Buffy coat protein, which have been activated by the NDV or the Sendai viruses, which acts as a good inducer. But ultimately, whatever the interferon that are produced from the other, they provide less immunity when compared to the species-specific interferon. So later on, with the development of the recombinant DNA technology, we now have the ability to produce more amount of human interferon by introducing the gene into either the bacteria or the yeast and cloning them to produce more amount of this interferons. And these interferons usually gives enhanced effect when they are applied to the local type of infection such as upper respiratory tract infections caused by the influenza or the hepatic keratitis, etc. But in case of systemic infections, they do not show much beneficial effect in the treatment aspect, and they are sometimes thought to be an anti-cancer agent. Recently it has found that these interferons are usually the regulatory protein and belongs to the family of cytokines. So initially it was thought to be antiviral agent, but now we know that it is not only antiviral but also anti-cancer, and they are a sort of cytokines. And the biological effect that the interferons usually shows is the first and foremost is to protect against the viral infection, that is antiviral effect. It provides it inhibits the infection of the different types of viruses. Next is antimicrobial effect. Apart from protecting infections from the viruses, it also have the ability to protect against the chlamydia, rickettsia, mycoplasmas, etc. Next is the cellular effect. It has the anti-proliferative activity like decreases the cell proliferation, or the DNA and the RNA synthesis, etc., are together combined in the cellular effect of the interferons. Next is the immunoregulatory effect. So as it is a cytokine, it also shows the regulation of the immune response by either activating or decreasing the T cells or the B cells or the natural killer cells, etc. So apart from being an antiviral agent, it also has antimicrobial activity, cellular activity, as well as the immunoregulatory effect. Coming to the lab diagnosis, how do we diagnose these viral infections? Initially, it was very difficult for us to identify or isolate the organism because it is an obligate intracellular parasite. But with the help of various rapid technologies, we now have a variety of techniques to identify or to diagnose the infections that are caused by the viruses, so that we can go with the successful therapy or the treatment aspect. So it is a diagnostic microbiology that helps us not only to identify the viral infection, but also helps us to know the strain by which the particular human host is infected by the viruses. So the best thing, or you can say the prophylactic measure to prevent the transmission of the infection, is the screening of an individual. In the case of the blood donors, you have to screen the individual to detect whether the blood is contaminated with HBV or HIV, etc. So this usually decreases the mortality rate up to 50% by screening the individual who are usually infected or when the blood is contaminated with this HBV or the HIV. Next is the etiological diagnosis of the viral infection helps us in the early therapy or the treatment aspect to prevent or the spread of the infection from one part of the body to the other part. Next is with the help of the lab diagnosis or the diagnostic virology, it is helpful for us to go with the early hepatic encephalitis treatment, and this also helps in identifying the vague syndromes that are caused by the organism and new syndromes that are usually produced by the organism due to the antigenic variations. Apart from that, you can also identify the epidemic and the antigenic variation. So whatever the treatment aspect is completely depend upon how successfully you are able to identify the virus and isolate the viruses to completely identify the strain which is involved in the pathogenesis of the infection. So the successful diagnosis not only depends upon how well you are able to identify, but how well you are able to collect the specimen. So it is completely depends upon the specimen, that how without contamination you are going to collect the specimen to give a good and a very accurate result of the viral infection. And now once the specimen is collected properly, it has to be labeled, it has to be transported, that we have seen in the diagnostic microbiology, and we should know the characteristic of the viruses, because there are certain viruses that cannot be refrigerated immediately. They usually gets inactivated. So one should have a very good knowledge, the lab technician should have a very good knowledge in collecting the specimen and handling the specimen before the lab diagnosis is usually done on the particular clinical specimen. So the tests that are done after the specimen is brought to the lab diagnosis is we go with a microscopy, demonstration of viral antigens, and isolation of the virus. So first and foremost is microscopy. Here we cannot make use of the light microscope because these organisms are ultra microscopic in nature. You have to go about with the most sophisticated technique, that is the electron microscopy, and identifying the virus elementary bodies. As we have seen in the inclusion bodies, the inclusion bodies can be stained, can be seen under the light microscope, but to identify the viruses you have to go with the electron microscopy. And the presence of the inclusion bodies, as we have seen, will help us to identify the different types of infection. As in the case of the inclusion bodies that are present in the nucleus helps in identifying the herpes infection, and the Negri bodies in the case of rabies infection. So these are certain aspects by which we can easily identify the organism with the help of microscopic technique. And the most sophisticated technique is we can go with the fluorescent antibody technique, which can be either applied as a positive or a negative antibody technique. You can identify, you can complex it with the viral antigen, and you can identify under the UV microscopy or a fluorescent microscopy. Next is identification of the lesions that are produced by the organism, can also be done either with the electron microscopy or a fluorescent antibody technique. It not only helps in identifying the viruses and the lesion, but also helps in identifying the viral antigens that are present in the viruses. Next is you can go with the demonstration of the viral antigens because it is antigen that are usually involved in the pathogenesis of the infection. So this viral antigen can be very well demonstrated with the help of serological methods, mostly by using the precipitation or immunofluorescence techniques. The other most sophisticated techniques are counter immunofluorescence or counter immunoelectrophoresis, radioimmunoassay, enzyme linked immunosorbent assay, etc. Apart from which, you can go for the molecular method, which helps in rapid diagnosis of the viral antigens. You can go with the PCR techniques or by using the probes. Next is the isolation of the virus. So as we know that these viruses cannot be easily grown in the artificial media. They require a living host as they are obligate intracellular parasite. So there was a difficulty in isolation of the virus initially, but now with the help of different techniques, we are able to isolate the viruses in the lab and identify the viral strain that is involved in the pathogenesis. So the specimen that has been collected has to be refrigerated immediately, because the organism cannot survive in the external environment, but at low storage or cold storage the organism can retain its viability, right? So you have to go for the refrigeration till it reaches the laboratory, where we can go for the isolation of the virus in the different types of cultures. So you can go with the animal inoculation, you can go with the cell cultures or the tissue culture techniques. And in order to identify the neutralization effect of this, you go with the antibodies that are present, which is determined usually by the serological methods. And the clinical data has to be given in the light of, that is, the result have to be usually given in the light of clinical data. And once the organism has been isolated, the immune response has to be determined before getting it confirmatory reports. Now coming to the serological diagnosis that is usually done for the virus are during the viral infection, you can see there is an increased production of the antibodies inside the human host. So identifying the titer of the antibody in paired sera helps us in identifying the viral infection. Paired sera means to say there can be two types of sera. One is an acute sera and the other one is a convalescent sera. In the acute sera, the sera that is immediately taken after the viral infection is called as the acute sera, whereas the convalescent sera is the one that is taken after 10 to 12 days. So you have to identify the paid sera to go for the confirmatory report with the serological diagnosis of the viral infection. Next, you can go with the neutralization effect of the antibodies, and the test that is done is a neutralization test or the complement fixation test. You can go with the ELISA and hemagglutination inhibition test, that is mostly done for the myxoviruses who expresses the hemagglutinin antigen on their surfaces. Apart from this, we have got more rapid techniques to identify the viral antigens or the antibodies. They are nothing but the molecular diagnosis using PCR or the probes. Coming to the immunoprophylaxis of the viral diseases. Now, the most important characteristic feature of these viral diseases is that they usually give a prolonged and effective immune response after the initial attack. So, the immunity is the most important thing in the case of the prophylaxis of the viral infection. So, this immunity can be enhanced by giving either the live vaccines or the killed vaccines that are prepared from the viruses. So, the first introduction of the vaccination procedure was done in the case of smallpox virus, which was given by Jenners using a cowpox vaccine. So we know the story behind how Jenner used a cowpox vaccine to protect against the smallpox. So that was the first method that was used in live vaccines usage to produce immunity against the smallpox. Now with the help of this, we have completely eradicated the smallpox infection. The other thing that was coincidentally discovered was the attenuated strain of the viruses in case of the yellow fever. So here the yellow fever vaccine was usually produced from the attenuated strain, which protects the immunity against the yellow fever viruses. And they are different methods, modified methods by which you can produce the vaccine from the attenuated strain. One such is the plague selection. This was used for the production of the Sabin vaccine against the polio. And apart from that, you can go with the TS mutants or the recombination technique to produce the vaccination for the influenza. So there have been many modifications that has come forward in producing both live and kill vaccine after the initial usage of the vaccine against the smallpox that was used by Jenners. Now coming to the importance of both live vaccines and the kill vaccines. The kill vaccines are usually the one that are produced from the viruses which have been completely inactivated or the killed. They are usually produced when exposed, the viruses are exposed to the UV radiation. But there are chances that by the use of UV radiation there can be a reactogenicity, that is the organism can get back to the normal form if they have not been completely killed. So there is a possibility that they might land up to cause the infections, and hence by the use of various chemicals we can completely kill the organism and can use them as kill vaccines. Apart from that, recently with the help of recombinant DNA technology we are now able to produce a subunit vaccine where the whole organism is not taken as such to prepare the vaccine, but the gene that is involved in the immunogenicity is usually taken, or the antigenicity is usually taken, and it is like cloned in the bacteria or the yeast, and then the vaccine are produced with the help of recombinant DNA technology. So that is called as subunit vaccines. And the example here which was very successful was the cloning of the hepatitis B vaccine or the hepatitis B gene to produce a vaccine against this particular infection. Now coming to the live vaccine, the advantage behind the live vaccine is you can give this live vaccine in the natural route through which the organism usually gain an entry and can provide immunity for a very longer period of time, or it can be throughout the life. So they provide a complete lifelong immunity. Apart from giving this, they can also enhance the cell mediated immunity, and they are more effective when compared to that of the kill vaccine and mostly used in the mass immunization. Coming to the disadvantage of this live vaccine is sometimes the immunity of the host can interfere with the vaccine that has been given. As a result of which there will be elimination of the vaccine that is given, which will not be able to protect the individual from the such infection. So the interference is one of the disadvantage of the use of live vaccines, and moreover they are heat labile in nature, and they can get easily inactivated when there is an increase in the temperature. And the complication that are associated with the live vaccine is sometimes the attenuated strain can get converted to the wild form and can cause the infection. Next, coming to the kill vaccine advantages, here there is no chance of the attenuated vaccine being converted to the natural wild type strain to cause the infection, because the organism is completely eliminated or killed over here, but it will retain the property of immunogenicity. And these kill vaccines are usually given along with the polyvalent vaccine or as triple vaccine in conjunction with the other type of vaccine to enhance the immunity against the different types of viral infection. But the disadvantage of the kill vaccine is because it cannot give you lifelong immunity, you have to go for the multiple booster doses for every 10 years or every year, depending upon the type of viral infection you want to protect with. So apart from these two vaccines, live vaccines and the kill vaccine, another important aspect of protecting against the viral infection is the passive immunization. So in case of passive immunization, as we know, there are two types of passive immunization, that is a natural passive immunization and artificial passive immunization. In case of natural passive immunization, the immunity is from the mother to the fetus, whereas in the case of artificial passive immunization, we go in for the antibodies that are present, that are produced in the other species to protect against the diseases. So passive immunization is mostly given to the non-immune individual, to the individual who is not able to produce its own immune response. So mostly in the case of measles, mumps, hepatitis, you can go for the passive immunization. Apart from that, a combined and a passive immunization has to be administered in order to completely prevent from that particular viral infection. Next and the last part of the host-viral interaction is the chemoprophylaxis and the chemotherapy. Now, apart from using the live or the kill vaccines, there have been many antibiotics that have been developed to cure the viral infection. So because these are obligate intracellular parasite, it becomes very difficult for us to target the virus because they are present inside the cell. So when we want to target the organism, we have to target even the host cell, so thereby causing more damage to the host cell apart from killing the viruses. But now we know that there are variety of sites on which we can make the antiviral substances or the chemotherapeutic substance to act upon only on the viruses without causing profuse damage to the host cell. So there are variety of sites where we can target and eliminate the viral infection. The first target is the attachment. So we can prepare the chemotherapeutic agent in such a way that the drug is not able to make the virus attach to the host cell. As a result of which, when there is no attachment, there is no initiation of the infection. Next target area where we can use the chemotherapeutic drugs are the transcription area, where the mRNA is not transcribed. And we can go with the translation area where no proteins are synthesized, and then replication, or you can target the assembly area, or you can go for the release of the viruses. So these are the different areas of target where different chemotherapeutic agents can go and interact and inhibit the viral infection. So apart from this, there are drugs that you can even target the cell-free virion, that is the virions which are present extracellularly. So we can even target these cell-free virions. And apart from that, you can go with the enzymes, specific enzymes that are produced by the organisms. Take for example the neuraminidase, or you can target the reverse transcriptase that are exclusively present on the virus. So chemotherapeutic agents can be prepared to target these areas to inhibit the viral infection, to eliminate the viral infection. And the first drug that was antiviral drug that was produced was in 1960, which is called as methazone, that was produced against the pox virus, and also against the eczema, vaccinatum, and the smallpox. In 1962, next important drug that came forward to be effective against the herpetic infection was the idoxuridine. Now later on, many broad spectrum of antibiotics were discovered. They were thought to be effective against the viral infection. A new molecule was developed recently, which is called as the amantadine, which is effective against the influenza A virus. And in 1970, acyclovir compounds were found to be effective against the herpes infections. So apart from these different drugs, we have now different antiviral agents that are able to target the different components of the organism and inhibit the viral infection. So we will classify them based upon the activity of these drugs on the viruses. So first, coming to the nucleoside analogues. Now these nucleoside analogues are nothing but the drugs that usually targets the genetic material of the viruses. So analogues are nothing but they are compounds which are which have the structural similarity with the basis of the genetic material, such as the adenine, guanine, cytosine, and the thymidine. So during the replication process, these analog usually goes and binds to the viral nucleic acid and completely changes the viral nucleic acid and inhibits the viral infection. So the best example here is a deoxyuridine, which is an analog of thymine, and it blocks the thymidine kinase and is highly effective against the herpetic keratitis. So this is said to be a bit toxic towards the host cell also. So a modified form of deoxyuridine called as 5-iodo-2-deoxyuridine was produced against the herpetic keratitis. So these are the analogs that usually targets the DNA of the host cell and prevents the viral infection. And another compound that was developed that is thought to be more stable when compared to the 2-deoxyuridine was 5-trifluoromethyl-2-deoxyuridine. It is more stable and more effective when compared to the idoxuridine that was initially prepared against the viral infection. Next is the bromovinyl deoxyuridine, the recently developed drug against the herpetic infection. And not only against this herpetic infection, this can be effective even against the varicella zoster viruses. Next group of chemotherapeutic agents are adenyne arabinoside. So in the case of this particular compound, there is arabinose in place of the ribose that usually attaches the nitrogen base. So there is a variation over here now. Now this variation, when it goes and attaches to the nucleic acid, it completely inactivates the nucleic acid. So adenine arabinoside is also a compound that was found to be effective against herpetic keratitis, and it was thought to be a bit cytotoxic in nature, and hence cytosine arabinoside was produced, which which is said to be less toxic when compared to that of the adenine arabinocyte. And to treat the systemic infections, a new compound has been developed which is called as the acyclovir, which is called as the acyl guanosine. It is a you can say an analog of the guanosine, which is effective against the herpes infection. And later on, many more acyclovir compounds were developed which are less toxic to the host cell and more effective towards the viruses, which are ganciclovirs, effective against the cytomegalovirus, azithromycin. This is the most important drug that is active against the HIV. Now, the action of the azithromycin is it prevents the formation of the pro-DNA acting upon the reverse transcriptase. Now, during the HIV infection, the virus usually has a single stranded RNA. Now, this RNA gets converted to a hybrid of RNA DNA, which later on gets converted to the double stranded DNA. Now, this double stranded DNA is infectious in nature and forms a latent virus. Now, the best way to target this HIV virus is to inhibit the replication process by acting upon the reverse transcriptase, and hence when there is no reverse transcriptase, there is no synthesis of the double stranded DNA. So azithromycin is the drug that usually acts upon this particular area and inhibits the HIV infections. So similar type of drugs were developed which are called as dideoxynucleosides, which are said to be anti-HIV in activity. Apart from that, you can target the protease activity of the HIV, which are called with the help of protease inhibitors called as ritonavir, etc. So these also have the ability to inhibit the viral infection by interfering with the replication of the viruses. Next compound is a ribavirin that is effective both against the DNA as well as the RNA viruses. Now, other drugs that can be used to inhibit the viral infections are amantadine A against the influenza virus, and amantadine and rimantadine are mostly effective against the myxoviruses. Rimantadine usually acts upon the neuraminidase and inhibits the neuraminidase activity. So, as told, the attachment is the first step and it can be inhibited by amantadine A. But with the help of rimantadine, you can prevent the release of the progeny virus. So both are effective against the influenza viruses. Next, we have enviroxin, which is effective against the rhinovirus, and foscarnet trisodium phosphonate, which is effective against wide variety of viruses such as herpes simplex, hepatitis B, and HIV. So till above we have seen that they are active only against one or two viruses, but with the help of foscarnet you can see the broad spectrum activity of this particular like chemotherapeutic agent. And the suramin is another important type of therapeutic agent that is active against the parasitic drug, that is called as the anti-parasitic drug. And in case of HIV infection, the target of the suramin is again the reverse transcriptase. So this is about the chemoprophylaxis of the viral disease.